Hi, welcome to the Truong group working on schizophrenia. This will be an overview of our fall semester, and presenting will be me, Cameron, as well as my research partners Khushi, Isha, Jia, Nithya, Ray, Alisha, Sadhana, and Kira. So our objective of this semester was to test clozapine, an atypical antipsychotic drug, on genetically modified C. elegans, specifically the E1370 strain, to create a predictive model on how clozapine will affect depressive symptoms on schizophrenic patients. So what is schizophrenia? Schizophrenia is a mental disorder that affects a person's cognition and senses. Normally, when you think of schizophrenia, you think of paranoid schizophrenia, but we're testing catatonic schizophrenia, and that's when a person shuts down mentally, emotionally, and physically, and exudes depressive symptoms.
The treatment we are testing in our research is clozapine, an atypical antipsychotic drug that targets both dopamine and serotonin receptors. It is mainly used to treat the symptoms found in schizophrenia and schizoaffective disorders. It does this by balancing the neurotransmitter levels in the brain. Some uncommon side effects include fatigue, weight gain, headaches, and more. These side effects can be heightened if alcohol is consumed.
Currently, we're practicing with wild typeworms, which are not genetically modified. We're doing this so we can perfect our procedures and establish a baseline as a control group before working with our transgenic or our genetically modified strains. We're observing, keeping account of, and taking pictures of these worms. On the right is a picture of the wild typeworm that we took within the past month. In addition, we've learned a lot about possible causes of contamination and how contamination can affect the worms.
Specific strains of C. elegans show phenotypes similar to the manifestation of schizophrenia in humans, so we use them to assess drug efficacy on schizophrenic symptoms. For our research, we will be testing the effects of clozapine on the schizophrenic symptoms of the E1370 C. elegans strain. The E1370 strain has a phenotype for diminished motivation due to the overproduction of serotonin neurotransmitters that are involved with motivation, and this is caused by a mutation they have in the DAF2 gene. Specifically, they have a functional loss in the mutant insulin receptor that corresponds to this gene, causing them to become immobile even when they experience acute food deprivation. Acute food deprivation would normally stimulate animals' foraging behavior to find food, so by remaining immobile, C. elegans show signs of being suicidal or depressed.
The synchronization process is an important part of our entire experiment. We use different buffers and a bleach solution to separate the eggs from the worms. The bleach harms the worms, but not the embryos, due to the eggshell protecting them. The main objective of this process is to get the worms at the same developmental stages. This is especially beneficial in performing assays because it creates no difference in the developmental factor of the worms.
Apart from our experimental strain groups, some other things we're testing is different dosages of clozapine. And so the way we do this is by currently adding it into their food source, OP50, which is a type of bacteria. In it, after placing it on the Petri dishes, we dry it with these different concentrations based off of a clozapine stock solution that we dilute with DMSO.
So in the future, we are planning on exposing these wild type worms to clozapine. This will allow for a control group, meaning a group of non-transgenic or non-mutated worms, to be observed and data recorded, which would provide us with baseline observations. This type of test would allow for us to get an idea of how clozapine affects C. elegans specifically that do not exhibit schizophrenic symptoms or behaviors.
For our future plans, we're having a cooperation with the chemistry group, and we will be looking into methods that measure how chemicals are transmitted between neurons in the worms and measuring how clozapine and other compounds affect this chemical transmission.
In the near future, we plan on testing clozapine on the E1370 strain. This strain becomes immobile when it faces acute food deprivation. However, the worms still have the ability to move in response to touch or exposure to aversive chemicals as they are not paralyzed. The normal behavior for C. elegans would be to forage for food, but this strain of worms remain in the same location on the plates until they die. This correlates to the avolition and apathy phenotype frequently seen in schizophrenic patients. When treated with clozapine, we expect to see the worms continue to move and forage for food despite their regular tendency to become immobile. Thank you for listening, and a special thanks to ASDRP for this opportunity and being willing to help, as well as Ms. Truong for guiding us every step of the way with their expertise.